Thank you all so much for staying. I have to bring that joke, like I knew that PacBio is a hot technology, but today they're overdoing it and demonstrating this. So I don't know why this looks a little bit as I've murdered someone, at least from my angle. But it's a great pleasure today to introduce to you our work that we did over the last couple of years together with PacBio and Twist together. In the same sense of what we just heard on the SIP2D6 panel, we extended this concept a little bit. So as far as I'm going to show you today, first I want to introduce a little bit about what are structure variants and why we do care about them, and then I'm going to dive into the matter a little bit more. Let's see if this works. So structure variants are loosely defined as 50 base pairs or larger alterations, and we can divide them into deletions, duplications, insertions, inversions, and translocations, and basically we identify them by looking at abnormal mappings of the reads to the reference genome. They play a key role in different aspects of medicine and evolution and other categories, such as gene duplications or gene deletions along the tree of life, genomic disorders. Here I'm showing a breast cancer cell line picture of all these different molecules that are forming, impact on regulation, and then impact on phenotypes, where you see here mapped out heritability contributions of structure variants in red. Red are copy numbers, black are rearrangements, and in gray you have SNPs. And so what I always like, and why I bring always these slides in my talk, is like only when we bring together structure variants, SNPs, and tandem repeats, and all these variants that are co-occurring in our genomes together, we can really improve the heritability in our understanding of certain genomic diseases. So this is really something that drives my lab a lot. And so over the last couple of years, we and others have really shown that long reads really improve the identification of structure variants, being it, for example, that short reads only detect around half of the variants that we can see with long reads, but also give us resolution in repetitive regions, especially also with medical relevant genes in repetitive regions that we sometimes just simply ignore because of their complexity in a lot of studies. And then all of us are used to these days about T2T genomes and assemblies and phasing, but it's good to remember that long reads really enable this as well and bring this to the table. So what I maybe should have chosen for the title today is really what is the utility for long reads for clinical or human research overall. So questions of like, well, do we actually need this for medical research? Are we good as is? Are there benefits for diagnosis, and how can we lower the costs and improve the utility of the long reads itself? So in terms of do we really need this for medical research, I think the answer is pretty straightforward. We have kind of a good list, around 400 genes, autosomal genes that we published in 2022, but also Mendeke et al. Published a list of 193 genes. There are really complex alleles in the human genome. Phasing improves the interpretation. And then there's also epigenetic that we honestly know so little about these days, but there are some quite promising results being published these days. Highlighting importance in Mendelian diseases and cancer, such as HLA silencing and so on and so forth. Are there benefits for diagnosis? Well, the short answer is kind of yes, but it's hard to assess, right? So what we often do, especially in the center or a lot of other groups as well, we're looking at genomic benchmarks and how good or bad these genomic benchmarks can be resolved with different technologies. So right now we have basically two big benchmarks available from GenomeBottle, which are the SV benchmark and the SNV benchmark. While the SNV benchmark actually integrates a lot of the genome, so like 92% of the genome, it still falls short in certain regions, such as SMN1 and 2 or other tandem repeats. The SV benchmark is actually much more focused because it's also like a little bit derived from short reads, so like there are certain disadvantages in this. But like what really worried me or what really was missing is like a really focus on medical regions. Like we can talk about intergenetic and unique regions and variants in there for ages, and this will drive our accuracy scores. But how do we really perform in these challenging medical relevant genes? And so for this, GenomeBottle and we set out to formulate a new benchmark, CMRG, which was assembly derived. So basically we took a hi-fi assembly, overlapped that with the existing benchmark to not kind of cope with the same regions again and really be unique. And then of course of these regions that were left over, we annotated them for the clinical relevance and we put that together. In this process, however, what was really interesting, and the postdoc of mine was working on this, is we found multiple errors in the reference that we could further on reduce. So this is GRCH38 having errors in around 30 or so protein coding genes and 12 or so medical relevant genes in the genome being falsely represented. For example, CBS is a good example where there is a copy of CBS falsely introduced in GRCH38, which leads to mapping artifacts and therefore not sufficient variant calling and so forth, so forth. So how can we fix this? A postdoc of mine, as I said, Sairam, worked on this publication, we call it Fix It Felix, to reduce the burden and then to speed up the correction of variant calling in your BEM file of interest for short reads, but also of long reads. So I just wanted to highlight this work from Sairam. The really interesting thing, however, and that drove a lot of other research in my group, was doing this benchmark activity. What we learned is actually that we just can identify or put out around 270, 260 genes of these 400 genes that we characterized as not overlapping with the previous benchmarks, but like being medically relevant. And 122 genes we completely missed out, which was interesting because like we didn't miss out because the assembly didn't characterize them. We basically missed out because like they were so complex that we couldn't compare them across technologies and across different variant callers or mappers. So this kind of really sparked a lot of other research in my lab of how to compare and tandem repeats and so on and so forth. But like the 400 genes that we characterized or tried to characterize in these benchmarks are also great because like they overlap a lot of different diseases, as you can see here on the right side. A lot of them are associated with neurological diseases, but like they have really profound impact in all other diseases. And I cannot stress enough how often we are missing out on these genes actually by doing standard Illumina genome sequencing, for example. So one of my favorite genes, for example, is LPA, just to demonstrate this. LPA is a cardiovascular disease important gene with a copy number of a 5.5 kb repeat motif that is repeated 7 to 50 times or more in human individuals. It's highly polymorphic. And we care about this because the copy number is directly related to the cardiovascular disease risk. There's an interesting thing to be learned about this gene because A, it's hard to assess and there are not many callers that can do it right now. But the other thing is like despite having different copy numbers in different ethnicities, for example, Caucasians versus African Americans, the cardiovascular disease is pretty similar. And so we are for sure missing a lot of variants that so far we cannot assess inside of this copy number. And then, I don't know if this laser pointer works, yes. So here, for example, we built together a graph based on long-read genomes of mapping out all the variations and across 40 individuals. And this is just across this copy number region. So like this region gets amplified up to 50 times per individual. And you see all these different paths in this graph, right? They represent different SNPs and different indels and different other variants. For example, here is a tandem repeat in the middle of the copy number variant. We are lacking tools right now to assess this, or for the probably impact and matter, because like there are also 2 exons present in this repeat regions itself. So with that in mind, we really require insights into these medical genes, not just LPA, but a lot of other medical genes that you're currently missing out a lot. And so that brings me, of course, to the main point: how can we lower the costs and improve actually utility of the long reads to assess exactly these genes, right? Lots of the other regions of the genome we can, in all frankness, do really well with Illumina because they are easy to map to and easy to assess. But how can we really kind of solve this remaining 400 gene puzzle efficiently? And so for trying to solve this, we set out in collaboration with Twist and PacBio together to create a panel which is around 22 MB large to really target these regions specifically and then get the most insights for the money spent, so to speak. So really scale up the resolution and scale up the insights that we can get in these genes. Overall, like you might note that I added like 3 genes to this panel, so it's 389 right now. We added GBA, for example, which is an important gene for Gaucher's disease and has an important recombinant, an important paralog close by. So how well or bad does this panel work? It's a pretty massive panel with 22 MB, right? So like, it's not easy to design this. And then again, like for Twist, we gave them really the hard regions of the genome that are medically relevant. So overall, we sequenced right now a set of 12 samples per review smart cell, and overall the results look really good. So like we're comparing here the panel data with 12 cells per review, compared to long read, hi-fi, one review smart cell, and then whole genome 40x Illumina data sets. So you see on the normalized coverage, they actually look really well. The panel is a little bit broader than the whole genome, but this is expected because like it's really focused on these medical relevant genes. Overall, the design failed on 4 genes of those, but again, like the panel actually captures them beautifully. The rest of the 385 genes, with 93% of all the base pairs of 8x or higher, which we determined sufficient for variant calling in these genes. And then here I just wanted to give you some insights of why we're actually doing this and why we actually care. So these are HIV screenshots where you can see each read is indicated by a gray bar. On top you always have PacBio, and the panel on the bottom you have always Illumina. And you can see we are actually really missing out. So like, I'm sorry I cannot read these genes from here on, and there is no image on the computer. But like you see clearly, like here we are missing out a good insertion that is only called with PacBio and not with Illumina itself. And then there is a much more complex region over here. Here's one of my favorite genes, GBA. You see here the gene, but also the pseudogene is captured because of the panel. So we're not just capturing the genes itself, but also the duplicated genes in the reference. And we can learn so much about recombination events between these different regions itself. So how do we analyze this data now? Well, Matt, my other postdoc, put together this pipeline where we used a modified GSH38 reference. So the modification is coming from Cyrum's work, from Fixit Felix, where we fixed these 12 medical relevant genes in GSH38. Then we do variant calling. This is an approach using mapping and assembly together. We identified the genes that really need assembly, and therefore we really need the time spent there. But also the genes that just can do with mapping alone. And then we have phased SNVs and indels together with phased SVs reporting. And these phase blocks are comparable. So like we did a lot of work to manage that the SNVs and the SNVs are in the same phase blocks. It sounds trivial, but it's actually not. And then in addition to that, we also run Bangu and Paraphrase, and then FarmCat actually to also look at SMN1, CYP2D6, NCF1C, and NI1P. So again, how good is it or how bad is it? This is just showing the fixed reference improvements itself. So here we are just aligning the panel to the original GSH38 and to our version of GSH38 that now is also publicly available from Genome Bottle and supported by GSEH. You can see based on the colors that we have a complete phasing throughout this gene, whereas like in the original 38, there's a lot of reads that are mapping across the duplicated region and this region here. And so we only can assess a few portions here for variant calling of this gene itself. And so therefore this fixed reference really, really matters. In terms of SNV calling, so this is again like the strong suit of Illumina. We compared it to Dragon, or GRTK, and pulled all the tricks that we can do. So you can see that the panel in blue, the whole genome hi-fi in red, and the Illumina genome in yellow are pretty comparable to each other in terms of performance across everything. SNV substitution calling, panel is on par with hi-fi, which outperforms a little bit of GRTK and Illumina. Indels, it's a little bit tricky because these regions are very repetitive, and so also the hi-fi approach sometimes stutters a little bit in these repetitive regions. However, the real benefit is really coming from tandem repeats and structure variants here, right? So you see in blue is the panel, red is the hi-fi whole genome again, and Illumina is in yellow. You see a clear performance difference. So here is the overall performance F-score between the different technologies. You see that the panel actually holds up to the PacBio whole genome, which is a fraction of the cost of the PacBio whole genome. And then clearly outperforms the Illumina reads itself. So we also looked at different replicates, because maybe this is just one run where we got lucky on HG002, and what we can learn from here. Overall, we see high consistency across the 3 different replicates of HG002. This is done by this plot. So how you read this is the data set is here on the axis, and then the dot represents for the category on top, where you have the list of variants. What is interesting to point out, in yellow and in green are variants, and the percentages are small, but it's hundreds of variants that were only base callable with hi-fi or with the panel itself. And then down here on the structure variants, similar picture. In yellow, only hi-fi whole genome or panel was able to call them, and overlaps with the benchmark from gene monobottle, compared to all of the technologies, for example, Illumina here as well. So I think it's pretty obvious, at least for HG00, this really brings something to the table across all 400 genes itself. So the last question, or one of the last questions, was really, can we actually improve the efficiency? So far I have showed you data from 12 samples per review smart cell, but like, can we reduce the costs further and really think about large-scale operations where we would deploy the panel, for example, in a TopMed manner or in an all of us manner, or like just on the side where you do your research or your clinical aspects about Illumina whole genomes? And the short answer is we can. So like, you see here the 100%, this is the 12 sampleplex, and you see that based on the y-axis, we don't lose much power until we get like at the 10% of the current coverage level. So this means we can actually, in theory, pool together around 48 samples on a review smart cell, putting everything together. And we ran rough costs, not with 48, but like with 35 to be a little more conservative per review smart cell. And this brings the cost down to $250 to $300 per run, all inclusive, including library prep and everything, which I think is really a compelling aspect compared to, I think, around $2,000 of a hi-fi genome. Depends on where you go. Oops, I did one too far. So besides these benchmark samples, we also were really interested in how it actually performs in non-cell lines, right? Cell lines are always easy. When it comes to tissues or blood, that's where the trick is really hard to get there. And so overall, we had like included 3 unsolved Mendelian trios, so in total 9 samples in a review smart cell, besides the other control samples. And we see a high consistency of Mendelian for SNVs as well as SVs with our pipeline, which is of course a good thing. We could find some interesting de novo candidates across the trios. However, upon closer investigations, those were unfortunately not the causative alleles for these trios. Furthermore, we looked at cardiovascular disease samples, and again here I'm looping back to the LPA gene that I just introduced in the beginning, which is really tricky cardiovascular gene. We have a little bit, I'm switching the buttons. We have a little bit of uneven coverage on the LPA gene itself, but you can see actually we can phase reads through the entire region of LPA, giving us much more in-depth resolution of different haplotypes and different haplotype-specific copy numbers and other mutations in this important gene, and therefore interpreting the LPA and cardiovascular disease risk per individual much better. We tried to do a simple correlation of coverage versus LDL, and like, yes, it's kind of significant. However, there are some outliers in here as well, so we are still investigating this. So to summarize my talk today, do we need this for medical research? I think absolutely. I've just shown you around 400 medical relevant genes that are otherwise often ignored, often missed. We're missing insights to them. We don't know much about them, to be honest, besides that they are relevant. Like, think about SMN1 and 2. We know that a certain exon is important, but that's about kind of it. We know LPA, the copy number, is important. There's so many variations behind them that we have so far been blind to, which is really, really important to get at. And now we have the tool not just to do it in a depth manner and really understand all the complexities and alleles, but also to scale it up, right? And to do it at cost. Other benefits from diagnosis, I think, again, like pretty clearly, yes. And then this panel really allows us to scale this up with, I think, around $250. Maybe we can optimize this more per sample overall. And so, yeah, with that, I want to thank all my lab members. I want to thank HGSC, PacBio, Twist, and Gene Monobottle for great collaborations, and on the right, some funding. Thank you so much.